So, good morning. Uh, I will talk. Oh, it's here. I will talk about the simulations that we are doing with some enzymes that break biomass to production of biofuels. This project, the PI of the project is Isaac Cann, and there's also a collaboration with Klaus Schulten in the University of Illinois. So the first generation biofuels, we used that already. Here in U.S., like the U.S. is the largest producer of ethanol in the world, and the cars can run, like most of the cars can run up to 10% of ethanol. We can also run on pure ethanol, like almost pure ethanol, 85% of ethanol, the E85. And the largest program that we know about biofuels, that we have today about biofuels, is in Brazil, and where no pure gasoline is found anymore. And the minimum percentage of ethanol in the gasoline is 18%, so between 18 and 25%. And where 97% of the cars sold in 2011 were what they call flex fuel, so they can run on pure gasoline to pure ethanol. You can choose whatever you want. But there is a problem with the competition with food production, because you're basically using food sources to produce ethanol, to produce fuel. So instead of producing food, you are using the corn to make ethanol or using the sugarcane to make ethanol. And for that, we have this, what we call the second generation biofuel. So instead of using the food sources, we are using the waste from the agriculture production to produce this more advanced biofuels. So the main point here is like we have to do enzymatic degradation of this biomass. And this part, like this enzymatic degradation, we know how to do. Like bacteria evolved this strategy to do that. But the main point is to reduce the cost, to make it profitable, to make it possible to produce today. How Blue Waters can help us to do that. And so basically what we have to do is simulations guiding the experiments. Most of the bacteria, they release the enzymes that can break biomass to the environment, and they capture their product back to use it. Like, so basically the products here, I mean, are the carbohydrates, like the short chains of carbohydrates. And the most efficient complex that we have to do that is not actually released from the bacteria, but is actually attached to the bacteria, and there's a huge complex of enzymes. They're called cellulosomes. So these cellulosomes, they were discovered about 30 years ago, but because of their very, very big and very complex, there is almost no clue about how they are so efficient. They are up to 50 times more efficient than free enzymes. And so that's why Blue Waters, because we need a huge computer, a huge system that we can simulate these huge complexes. And until machines the size of Blue Waters, it was just impossible. So what are these cellulosomes then? So cellulosomes are big complexes that you can see here. They have a big scaffolding that we call, and several enzymes here in blue. So the scaffolding in red and the enzymes in blue. And so here's the bacterial wall. So they attach to the bacterial wall, and they have these like arms that go to bring biomass close to the bacteria. This one that you're seeing here is the smallest cellulosome that we know. It's not the most important, that's the most common. So like in the same organism, the Clostridium thermocellum, the most common one have 63 enzymes. This one have only 9. And the most complex found so far, it's from this other organism, Rubinococcus flavifensis. It has about 200 different enzymes. And the main building blocks, so how they are built, like the main building blocks, so we have the enzymes that are about the same that we have in the free bacteria, but here they are attached to the scaffolding through these domains here, the dockerins and cohesins. So you have several of these building blocks it's like a huge Lego, like so you're building everything together with these enzymes. So to build an entire model for the cellulosome, we have to study the parts of the cellulosome. So we are studying enzymes, we are studying this interaction between these dockerins and cohesins, these building blocks, and also this scaffolding. Studying this dockerin-cohesin interaction, we started a collaboration with the Hermann Graub group in Germany, and Ed Bayer, that was the father of the cellulosome, who discovered the cellulosomes about 30 years ago, and Michael Nash, also in Germany. And we starts to study how these dockerines and cohesins, so the dockerine here, the cohesin here in green, and the dockerine in red, how they interact to each other. It's not a covalent bond, so it's a very strong interaction between two proteins. And what they observed in the experiment, so they did a single molecule experiment using AFM. They are trying to, so they are pulling them apart, trying to break the contact. And what they observe is that the forces that they measure is the highest forces ever measured for a protein-protein interaction. So it's about 4 times the force that you have in muscle cells, in proteins from your muscle cells. And so there is no other protein that gets close to that, as far as we know, and that's about half of the strength of a covalent bond. So first, they didn't know why that, and second, they observed two force classes, so 2 types of forces, one pretty strong and another one weaker. So the idea of the simulation was first to try to understand why you have two classes of forces. And then we did a simulation where we observed that if you bind the cohesin in the dockerine, sorry. Yes. Yes, that's the physical experiment. So we are trying now to reproduce that using the computer. And one of the ideas they have is that you can have an inverted mode, because all these enzymes are very symmetrical. So maybe what you have is like you have, instead of binding like that, you're inverting your protein interaction. And we try to build that. So using the crystal that was available in red here, the standard binding mode we created the inverted binding mode, we calculate both. And the rupture force for the inverted is much smaller, and in some cases we see something like that, like an unfolding of the system. That agrees pretty well with the results they have in the experiment. And then we observe that why that mode is stable, because we can, this is quite not so easy to understand if you first look, but so in the middle here we have the cohesin, and these are like lines of amino acids because we have helixes. And when you try to like put one domain to the other, they connect pretty well. And when you try to invert them, they also connect pretty well. So if you look, the characteristics of the residues, so hydrophilic interact with hydrophilic, hydrophobic with hydrophobic, so they connect pretty well. So you can build an inverted mode. And so basically what we did is like to pull them apart, just for you to have an idea. So you're pulling him the green and fixing the orange part, so and then we can try to break the system. Until we can, basically you can see like we start to make stronger and stronger and until we rupture the system. The forces that we measure are a little bit higher than the forces that they measure in the experiment, but that's normal because we are much faster than they are in the experiment. But the profiles that we observed and all the simulations are pretty similar. So one thing, why they are observing these extreme high forces then? So we kind of figured out, okay, the inverted mode is the one responsible for these 2 force classes. But why the extremely high forces? We observed that before like when we run the simulation without the forces applied, we see this interaction between dockerine and cohesin like that. You can see it's pretty loose. Like you can easily make this bond and break this bond. But after the forces applied, the system interacts much more. It's much stronger interaction. So it's like a finger trap. When you try to pull them apart, they interact more. And that's pretty clear when we analyze the area, the contact area between the 2 groups. So the contact area here, I'm just showing the residues, and here I have the residues. Then you can see what residue and what is the area of contact. So here we are measuring the area in contact with the other domain. And then you can see that unloaded, so before the force and loaded with the force applied, we have several cases where in the loaded we have like more, the contact area it's larger than the contact area before the force is applied. So when you try to pull them apart, they get closer. That's basically what we see. And and we believe that that's what the experimentalist calls of a catch bond. So it's basically one of the groups is catching the other, so and making the connection pretty strong. And okay, so just another comment. Here in the corner, you can see that the entire area, not separated per residue, but the entire area is also increasing when it's loaded and unloaded. And with that, we can create mutations and give the ideas for the experimentalists to try to make a different Docker and cohesion interaction complex. So now we have an idea about the Docker and cohesion interaction. We also have some ideas about cellulase that I didn't show here, like the enzymes, and we are trying to build the entire cellulosome. So together with the same group and other collaborators, we are developing an entire cellulosome model based on the smallest cellulosome we know. So we're using this SDBA domain, this scaffolding, and enzymes. So here are just some information for somebody that can understand about the system. Like these are the PDBs we use. So we have structure for most of the domains, but they have several very flexible linkers. So we are using this tool that we developed in the past. We are implementing that on Blue Waters now, and trying to use this tool to get new structures. So basically, this tool is a stochastic way to search for conformations, and we can use that like to fold proteins. So small peptides we can fold pretty well. So the idea now is to use the same tool to study like thousands of millions of conformations of the same cellulosome, and then do a cluster analysis, what we call, so separate them in the structures that are similar and understand how the motions of this huge system can happen. And so this is the first model that we got using this strategy. This is the first model, like atomistic model of cellulosome as far as we know. So this is the SDBA domain. This is the part that's attached to the bacteria. This is the scaffolding. And these are the enzymatic domains. So this complex has about 13 million atoms when you include water. And so this is a very short simulation that we did to see how stable the system was. Like this is just like the beginning of, we have to do that for several structures now. And with that, because we want to study how these complexes can reach to the biomass. So just to have you an idea, like this part is attached to the cell, as I said. And so when you don't have biomass close to the cellulosomes, in general, they are packed close to the cell wall. But when you have biomass, they can stretch and go to the biomass. So we want to understand how this stretching mechanism happens. And that's it. Thank you to Blue Waters for the computer time, and my collaborators in Munich, in Israel, and here at the University of Illinois. And thank you for listening. Questions?